Okay, before we dive in, let's kick things off with a massive question. How do doctors actually know what treatments work? I mean, it's not just, educated guesswork. It's an absolutely rigorous, highly structured mathematical and scientific process. Welcome to This Explainer. Today, we're basically putting on our detective hats to reverse engineer the absolute bedrock of medical research. We're going to look at exactly how healthcare science manages to draw these massive, life-saving conclusions from highly specific, carefully chosen pools of data. Here's our roadmap for today. We'll hit evidence-based practice, study designs, look at a really fascinating case study on self-reporting, and then wrap up with populations, samples, and sampling errors. Let's get right into it.
Section 1. Evidence-based practice, the foundation of healthcare.
Think of evidence-based practice, or EBP, as this perfect, precise fusion of three crucial elements. You see, clinical decisions are literally never made in a vacuum. They exist right at the intersection of the best research evidence, the provider's own clinical expertise, and, of course, the individual patient's values. You absolutely need all three of those to make conscientious, explicit, and judicious decisions about care. And the whole aim of quantitative research in healthcare is to find that best evidence objectively, so we have a reliable foundation to stand on.
Section 2. Designing clinical research studies, experimental versus observational.
So how do we actually gather that best research evidence? Well, we basically have two main tools in our toolkit. On the left, we've got experimental studies. This is where researchers deliberately step in and influence events to see what happens. A prime example is a randomized controlled trial, or RCT, where patients are randomly assigned to different treatments so we can compare the results. Now, on the right, we have observational studies. Here, there is zero intervention. Researchers are just watching. They simply observe and record behaviors, symptoms, or attitudes as they naturally happen. Cohort studies, which track a specific group of people over time, fit right into this category.
Section 3. Case Study. Self-report accuracy. The West of Scotland investigation.
To stop this all from feeling too abstract, let's look at a really fascinating observational study, a cross-sectional one actually, conducted in the west of Scotland back in 2004. For context, we know that smoking during pregnancy heavily increases the risks of severe complications like premature birth and sudden infant death syndrome. Researchers wanted to check the accuracy of self-reported smoking status during pregnancy. Because antenatal screening relies heavily on women just telling the truth about their habits, researchers suspected that immense media and social pressure might cause some women to, underreport their smoking. So they selected 3,475 pregnant women at their second trimester screening to find out what was really going on.
Now, this is where it gets really interesting, looking at the human element compared to the hard biological data. When directly asked, 24.1% of the women identified as current smokers. But when researchers actually checked the biological data using a catenin analysis, which is a test where a specific chemical concentration definitively proves someone is a smoker, the true number was 30.1%. That is a full 6% discrepancy. Self-reporting literally underestimated the reality of the situation.
Section 4. Populations, samples, and estimates. The mechanics of research.
This whole case study perfectly represents the core rationale for sampling. See that large circle on the left? That's our population, which could be theoretically infinite or just geographically massive. Because it's literally impossible to physically test every single person to find fixed population parameters, researchers have to take a smaller chunk, a sample. This sample gives us what we call a point estimate, and researchers then use that estimate to make an inference backward, trying to understand the entire population as a whole.
But you've got to ask yourself a vital question here. Can a sample of 3,475 Scottish women truly be generalized to the rest of the UK, or even the world? The answer to that heavily depends on the characteristics of the sample itself, and that introduces a very, very important statistical concept.
Section 5. Understanding sampling errors, why samples aren't perfect.
Now, sampling error sounds bad, but it isn't actually a mistake per se. It's just the natural magnitude of difference between our sample's estimate and the true population. Think of it like arrows hitting a target. They aren't all going to hit the absolute dead center, right? Because we're estimating from just a single sample, there is a built-in theoretical uncertainty, simply because we can't quantify the entire population perfectly. We often represent this uncertainty later down the line using something called 95% confidence intervals. To try and navigate this error, researchers use a few different approaches to sampling. Random sampling is the gold standard. It ensures everyone has the exact same probability of being selected. That's great, but honestly, due to time and money constraints, it's rarely used across a whole population. Then there's cluster sampling, which focuses on natural groups, like, say, inviting all the patients within a few randomly selected hospitals to partake. And finally, convenience sampling, which brings us right back to our Scotland case study.
Convenience sampling is exactly what it sounds like. Selecting participants based on ease of access for the researchers, like picking a single clinic or one specific geographical area, rather than using true randomness. Our Scottish smoking case study actually used convenience sampling. They pulled their data from specific clinic registers right there in the west of Scotland. Now, the women were selected at random from those specific registers, sure, but because the study was restricted to that one specific location, it wasn't perfectly random across the whole population.
So, the big takeaway here is that the result you get is solely tied to your sample. Is your sample truly representative? Well, that's impacted by a bunch of factors. Your sample size, how, where, and when the sampling actually happened, the specific characteristics of your participants who flat out refuses to participate, and of course, the accuracy of your measurements. I mean, just look at that 6% difference we saw earlier between simply asking someone if they smoke versus taking a hard biological coteening measurement.
As we wrap up this explainer, I really want to leave you with a final empowering thought to apply to all the medical news you encounter every single day. The next time you read a flashy medical headline claiming a huge new breakthrough, ask yourself, who was actually in their sample? And is that sample truly representative of the real world? Keep questioning the data, guys, and thank you so much for joining me on this explainer.